Okay, good morning everyone. So yesterday Rafa brought us all the way from our images to our validated cryo-EM construction, and today Alan is going to tell us how you built an atomic model inside.
Okay, thank you. So back in the early days of Cryo-EM, interpreting a map was really easy. You just squinted your eyes and described the features of the map that looked like the human body. And if you were British and found that too personal, to a teapot, things that we were comfortable with. And that's why in structural biology, terms like handle, spout, head, arm, body, knob dominate. However, nowadays, things aren't so straightforward. And simply pointing out the bits of your map that looks like a phallic appendage, although it's still funny, is no longer suitable. And at resolutions better than 4 angstroms, we're expected to provide an all-atom model. And interpreting a map with an all-atom model typically requires 3 steps: model building, refinement, and validation. And my talk will pretty much follow these 3 steps sequentially, but in real life, where some of us live, these 3 steps are interrelated and often happen simultaneously. Now, because there's a limited amount of time, I'll really just focus on tools that are available from these 3 packages: CCPM, Phoenix, and Rosetta. There's lots of other good tools out there, but really the ones concentrated in these packages are very good. But before we start talking about the actual initial process of how to build a model, let's just consider the maps that we use for model building. So as Rafa mentioned, at the end of the processing stage, you generate a final post-processed map. And this map tends to have a single B factor applied to it. And in Relion, this is calculated by automated means through methods proposed by Peter Rosenthal and Richard Henderson. But this single B factor sharpened map may not necessarily be the best map for model building and refinement into. Now, if we have a look at one of the deposited maps, this is for the 2.2 angstrom structure of beta-galactosidase, we can see that the deposited map is actually quite noisy. And this noise here is a kind of an example of when a map is over-sharpened. So we have lots of high-resolution features, but we also have noise. If we start to blur this map, you can see that the noise starts to disappear while some of the high-resolution features are maintained. And it now becomes a much nicer map to build into. If we continue to blur the map, we start to lose some of those high-resolution features. And so there's clearly an optimal value. Now, in the CCPM package, there is a program called MRC to MTZ, which actually uses RefMac. And in this program, you can sharpen your final map or blur your map by user-defined values, and then you can plot them like this, which plots the structure factor amplitudes against resolution. And you can see for the deposited map, because the high-resolution information is kind of overweighted, it never tends to 0 at high resolutions. But really, we know that the amplitudes should go towards 0. And so maybe a map that's been blurred by a factor of 40 is a more suitable map to build into. So if we just again have a look at the deposited map, you can see this part of the model, which seems to be in broken density. There doesn't really seem to be any density for it. And this broken density is another kind of feature of over-sharpened maps. But if we were to blur that map by a factor of 40, you can now clearly see where that particular model should fit. And you could probably make a better model in this part of the map. Now, Tom Terwilliger, in part of the Phoenix package, is trying to come up with automated methods to blur your map in a way that maximizes the information there and facilitates model building. And his program it aims to do two things, which is to maximize the surface area, which is the kind of proxy for high resolution information, and then minimize the number of kind of contiguous regions. And by doing that, it tries to obtain the best B factor. But it's clear that we really need local B factors, because one B factor is not suitable for the whole map. And regions at the periphery will need perhaps, say, more blurring than a better well-defined core. Now, in X-ray crystallography, which is a kind of historical method of structure determination, map manipulations are common. And this is really a requirement necessary because of the way in which the phase information is generated. But these maps should not really be used for cryo-EM. There's a substantial risk of introducing model bias. And really, any new method of map improvement needs to be very rigorously tested to make sure it's not introducing model bias. And the safest approach is really to go back to the original maps. And Garib Murshidov at the LMB has written a paper in Methods in Enzymology, which discusses some of the potential misuse of crystallographic maps and techniques in cryo-EM. So once we're happy with our map, we start to need to think about the model. So if your structure exists in the PDB and you'll just obtain it from the PDB, maybe you want to first of all think about re-refining that model against the original data, and maybe high-resolution crystal data, and you can do that either manually or PDB redo. And the purpose of this is to avoid propagating errors from the original crystal structure through into your EM model. If your structure doesn't already exist in the PDB, but there are homologous structures, then you can use template-based modeling to generate models for that. And at the bottom, I've just placed a few programs that I personally find useful to do that. Now, if there's nothing like it in the PDB, first of all, check, because most folds have now been solved. So is yours really a novel fold? Is there really nothing like it? If there isn't anything like it, then you can do ab initio structure predictions. There's programs for that. And also, you can generate what tends to be very accurate secondary structure predictions, and these are incredibly useful for model building. Now, also in cryo-EM, sometimes you can get a map and you have absolutely no idea what is in it. So this is some unpublished data where we isolated a native complex, and really for parts of the model we had, or parts of the map, we had no idea what was in there. And so we developed this pipeline actually for a previous project, which was to take the map or sections of the map and then search in that map everything in the PDB. Well, you don't really want to do everything in the PDB because that would take a long time. So the Bulbez database is a database of all the high-resolution unique domains present in the PDB. And so you can search the map for this whole library of PDBs and identify proteins that most closely resemble your map. And so in this case, we identified, bizarrely, an acyl carrier protein, which really fits the map. But regardless of your starting model, you will need to do some kind of model building. And this model building can be either automated or manual. So in automated model building, the idea is you take your map, you click a button on the computer, and it generates a model. And the reason for doing this is that humans are lazy. We have other constraints on our time, like tea break and children. And also humans make errors of judgment, like having children. And so automated methods can be a godsend. And there's really 3 current methods that are available to do automated model building: Buccaneer, Phoenix Map to Model, and Rosetta. And I'll just discuss each of these in turn. So Buccaneer is developed by Kevin Kaltan at the University of York. And the idea is to trace protein structures in density maps by identifying connected alpha carbon positions. And to do this, it takes a known reference structure, And then it simulates a map from that based on your experimental map, taking into account things like resolution. And from that, it then identifies features that it can then use as a likelihood target to search in your experimental map. To illustrate this, we'll take the very first step of Buccaneer, which is to identify candidate C alpha positions. So it takes this simulated map from the reference structure and it superposes all the C alpha positions in that. And from that, it calculates a mean density and also a variance density. And this together becomes the likelihood function that you can then search in your experimental map to find all possible candidate C alpha positions. Because C alpha positions are kind of spaced evenly, it then becomes possible to then connect the C alpha positions by kind of lateral growing in a way that can then extend the chain. And then you start to merge fragments and link termini. And to sequence the chains, you simply look at the C beta environment and you do a likelihood comparison between the density for that residue in your experimental map with the density, average density obtained from the simulated reference structure map. And that allows you to assign the side chains. Now, Phoenix Map to Model, again developed by Tom Terwilliger as part of the Phoenix package, allows you to build not just proteins, but also nucleic acids. Currently, it's only available through the command line. And it starts off by doing some simple kind of map modifications to simplify the model building. So it first of all shrinks your box until it's just around your map, and then it tries to segment out if you have any kind of symmetry or into individual proteins, and then it does the automated kind of map sharpening, and then it does the actual model building. There seems to be multiple algorithms to do the model building, but the method is not yet published. But one of the methods it uses is Resolve. And Resolve is an old program developed for interpreting crystallographic maps. And the idea here is that rather than use individual atoms, it uses larger fragments. And larger fragments are more robust. And so you can potentially build at lower resolution. And it starts off by identifying helices and strands. So it has a density template for a 6 amino acid helix and a 4 residue beta strands, and then it searches for these in the map. And once it's found these particular elements, it then superposes on those a library of helices and a library of strands, and it's looking for the longest fragment that matches the density. So once it's placed the secondary structure elements, it starts to grow these, and it grows these by adding tripeptide fragments to both the N and the C termini, always looking for which fragment fits best to the density. But it's not just looking for how well it fits the density, but it's also looking to see whether if you place that fragment there, can I subsequently build the next fragment? The side chains are assigned by this kind of probabilistic alignment method, where you look at the probability of an amino acid being in that position given the density and also given the composition of the protein or nucleic acid chain that you're trying to build. So Rosetta is a relatively new method for cryo-EM, but the actual package, the Rosetta package, was first developed in David Baker's group, and it has a number of algorithms for protein structure prediction and designing novel folds and such like. But it's really been Frank DeMal's group who has modified this package to do structure determination from cryo-EM maps. And the way that this works is instead of using a predefined library like Resolve, it takes your specific protein sequence, and from that it generates fragments from the PDB that look similar in sequence and similar in predicted secondary structure. And then it performs a 6-dimensional search of these fragments into the density map. And it scores the fragment by this function that includes 4 terms, which is how well that fragment fits the density. Does the algorithm place the same residue in the same position more often? So that's the overlap score. A closability score are fragments that are close together in sequence, placed close together in space, and then it excludes fragments that overlap and clash. It then performs Monte Carlo sampling to obtain the maximally consistent subset of these fragments, and then assembles a often incomplete model. It then masks a map based on this model and then iterates and repeats the search with a new fragment library. And this approach requires that you can build up to 70% of the sequence. And once 70% of the sequence has been filled with these fragments, it then uses the comparative modeling tools in Rosetta just to fill in all the gaps. But the problem was that you did need to get that 70% complete model in order to progress to this stage. So just last week, they published an update to Rosetta called Rosetta Enumerative Sampling. And a bit like Resolve, it says, well, once you know some of the model, you don't necessarily need to do this exhaustive 6-dimensional search. You know where the fragments will be in space. And so you can take your incomplete starting model, and then you can design fragments for the sections that are missing, and then add those fragments one residue at a time. And while it does this, it's always keeping not just one option, but many options. And then these options keep getting pruned as you continue to add new fragments until eventually you've completed the loop or the section of density that was missing a model. Now, many of these strategies are actually what we do manually, whether consciously or subconsciously. And regardless of how well your automated model building works, you will need to do some form of manual model building or model adjustment. And really the standard package for doing that is COOT, designed by the LMB's own Paul Elmsley. And this really is a comprehensive package for model building and also for refinement. So it has a number of great tools, and this is one of my personal favorites, which is JiggleFit. And it's designed for when you've kind of placed something in density. You know it kind of fits this density, but you're not quite sure how. And this can be a whole molecule, a protein chain, or just a small molecule. And it starts by applying a random set of rotations and translations. It then scores these relative to the density, selects the top 20 solutions, does a rigid body refinement on each of these solutions, scores again, and then that finds the best hit. And it works incredibly well when you more or less know where something fits. But if you haven't got a domain or some kind of model to fit in there, by far the best way of starting de novo model building is to place secondary structure elements in the same way that we saw Resolve working. And helices especially, they're very well defined in the density, and placing helices in COOT in conjunction with JiggleFit can find the correct orientation of helices almost 100% of the time. So this gives you not only a starting place to build your model, but it also tells you the orientation of the chain that you want to build. And in COOT, you can either add a helix in a particular bit of density, or there's an automated method just to populate the entire map with helices and strands. But helix identification does work the best. And then once you have these secondary structure elements positioned, then you want to start building from those, again, like how Rosetta builds. But you can either build, say, one residue at a time, and by default, you can add an alanine, or you can assign a sequence to an incomplete model, and then when you start to add the terminal residues, the terminal residue will be instantly the one that comes next in your sequence. But if you're just adding polyalanine tracers and you want to sequence those, it really helps to know your density. And this is a really good example from, I think this is Wild Chu's group, where they've just plotted some density examples for each of the amino acids. And this is more or less what those residues often look like in about 3 angstrom resolution EM maps. So in the top left-hand corner, you can see that for the negatively charged amino acids, we're often missing density for the carboxylate groups. And this is because of the way these negatively charged groups scatter electrons and also undergo kind of radiation damage. And so these kind of knowing your density can really help you to build and to assign sequence to your models. If you didn't want to build one residue nucleotide at a time, you can use this C-alpha baton mode in Qt, which starts off by taking your density and then converting that into a skeleton based on where the density is highest. You can then trace this skeleton along, placing C-alphas at closely spaced intervals. And then you can just take the C-alpha trace, convert that into a main chain, and then assign a sequence to that main chain. And building nucleic acids follows the same kind of principles. You can start off by introducing ideal sections of DNA and RNA. You can then extend from those one nucleotide at a time, or you can use R-crane, which is analogous to this C-alpha baton mode, where you try to follow the phosphate positions to build the nucleotides. But unfortunately, the phosphate positions are often not that well resolved in EM maps, certainly not as well resolved as in crystallographic maps. And so R-crane doesn't work as well as it does with crystallographic maps. So once you've built your model, then or often as you are building your model, you want to start to move these atoms around. And Qt has many tools to do this. So you can do a real space refinement. This can be along a chain, or it can be in a sphere around a particular point. You can regularize that chain. You can do rigid body movements. You can rotate, translate. You can fit rotamers. You can edit torsion angles. And pretty much anything that can be moved, you can move in Qt. However, one of the important things to mention is that although we often talk about high-resolution EM maps, they're really low resolution. And so it helps to use as many restraints as possible during model building. And this will prevent the introduction of errors. But some of these restraints, and I'll talk more about restraints later, only some of these restraints are turned on by default in Qt. So it's a good idea just to check and turn on the restraints which are applicable to you. And as you're adjusting these atoms in Qt, you often get you get live feedback, often in this kind of traffic light system, where good sections, like good bond angles, good bond lengths appear green, bad ones appear red. And so you're getting this live feedback. And also here, this is relatively new, which is where you have rotamers. They're colored in these different polygons, which gives you an idea of where you may have some rotamers that aren't quite correct. So this is a relatively new program from Tristan Kroll, who's now at the CMIR at Cambridge, called Isolt. And the idea behind this is that you as a user can directly interact with a molecular dynamics-based simulation. And really, it's particularly good on problematic regions, whether those are loops or things in not particularly good density. So you can select a loop, and then it takes into consideration not only that loop, but the surrounding environment, and then does a molecular dynamics simulation on these, and you can then interact with this molecular dynamics simulation. And one way that you can do that is through this haptic interface. And the idea is that you can then pull on a particular atom, and it will be harder to pull on an atom into a region where it doesn't want to go. I think the idea behind this is to make model building feel more like manual labor, real work. So there are some conundrums with model building. One is what to do about atoms that have no density. Do you build those loops where there's only weak or faint or no density? What do you do with side chains, which, again, have no density? Do you truncate them? And also, is one model enough? So cryo-EM is an averaging technique. And so our maps that we obtain will naturally be a result of averaging many different conformations. So is a single model really appropriate for that map? Or do we need ensemble of models? And I have no answer to these conundrums. The debate has been raging for many years in X-ray crystallography, and really it's up to the particular model builder to decide how they interpret these questions. So once you've built your particular model, then you need to do some form of refinement. And the purpose of the refinement is to improve the fit of your model to the density, and also to ensure that your model fits with prior knowledge, what we know about kind of protein structures, what we know about chemistry. Fitting to density is actually really easy, but maintaining stereochemistry while you do that fitting requires a kind of delicate balance. So what changes during model refinement are the coordinates x, y, and z, and the B factors, which are usually interpreted as a measure of the amount of motion a particular atom or group of atoms is undergoing. And then we can also refine, often at higher resolution, is multiple occupancies, where we see side chains in different conformations. But unlike X-ray crystallography, the map stays the same. Although in Rosetta, you can, as you also refine the model, refine the magnification factor applied to the map. So the map will change size, but the map itself, the phases, amplitudes, remain the same. And during refinement, we really do want to take into account as much information, as much knowledge that we possibly can. And we do that in the form of restraints. And restraints act to stabilize refinement. They reduce the chance of overfitting, and they make sure that the final model is consistent with what we expect. Now, many of the refinement packages contain a number of restraints, a standard like how atoms should be bonded together. And we know fairly accurately, the distance between two carbon atoms. And also, we can restrain the B factors so that atoms that are close to each other don't have wildly different B factors. If you have symmetry in your molecule, you can apply both symmetry constraints, which is when you think that each of the models, each of the monomers in your oligomer should be identical, and you can apply restraints for when you expect there to be slight differences between those models, but you want to really minimize those differences. And then we have external restraints. So these are additional restraints that can be added together with the standard restraints available in the refinement programs. And there's many different packages to generate these external restraints. So in CCPM, you have ProSmart for protein kind of restraints, libg for nucleic acid restraints, and in Phoenix, you have the secondary structure restraints, which generates restraints for both types of macromolecule. But these programs are often integrated with the refinement programs themselves. And then for ligands, which I'm not going to talk about, you have ACE drug from Fei Long and then Phoenix Elbow. So the idea is that we can use reference restraints. So you can take your target molecule, which may be a very poorly built manual model, and based on homologous structures what that structure should really look like. And you can take restraints from that reference structure in the form of distance restraints, and you can apply that to your target molecule to generate a better refined molecule. So essentially, we know in the homologous structure that atoms X and Y are close together. Therefore, maybe in our model, X and Y should also be close together. And we can visualize all these restraints in Coot, showing how, again, refinement is integrated with model building. So here you have two chains, and then the restraints colored in color based on how they compare between the reference structure and the actual target structure. And we can also apply secondary structure restraints. So we know how hydrogen bonds should be connected in beta sheets and how they should be connected in helices. And so we can apply restraints to make helices appear more helical and sheets to appear more sheety. That was terrible. That was awful. Sheet-like. Okay. And the same is true for nucleic acids. We know that the Watson-Crick edges of the bases should face each other, and the hydrogen bonds which should be a certain distance apart. So we can again include that information as these base pair restraints. So you can see in purple, without those restraints, it may try to fit itself to density in the optimal way, but by adding the restraints, it fits in a way that is consistent with hydrogen bonding with the opposite nucleotide. Also, we know that many aromatic residues and nucleotides should stack in a planar way together, so we can also include that information in these parallel plane restraints. So here shows a refinement of a phenylalanine, which is stacking with a nucleotide. If you do not include these restraints, refinement pushes it away from preventing it from clashing. But only when you include the restraints can you get nice stacking between the two. And these restraints are common for each of the refinement programs. They all use restraints. And really, there's 3 main refinement packages available. There's RefMac, Phoenix Real Space Refine, and Rosetta. So I'll just talk about each of these. So RefMac is developed at the LMB by Garib, and it uses maximum likelihood to refine the model into the map, and it refines the model in Fourier space. So this requires that you take your map and modify it to structure factors. But this is all done internally in the program. And for this and all the other programs, you simply input your MRC map that comes out of refinement in Reliant or whatever package you're using. Phoenix Real Space Refine, on the other hand, operates in real space. So you just input your map and there's no modifications to that. And although these programs operate in 2 different spaces, whether that's Fourier or real space, they actually have many features in common. But they also have some distinct features. For example, Phoenix can do a simulated annealing step, whereas RefMac can do composite refinement. And so where you take maps and you combine it and refine against those combined maps. So there's many different features to these packages, and you really perhaps want to consider exactly what you want to do, or maybe try both when choosing which refinement program to use. And then you also have Rosetta. So Rosetta rebuilds your model using this fragment-based sampling. And the idea here is that models that have been built by hand generally fit the density quite well. But often, in order to fit the density, you've had to strain the model to kind of push the chain into that particular density. And so it takes and it looks for regions of your model that doesn't fit the density well, and also for regions where there's local strain. And then it designs fragments, again, from known structures that could replace the section that you have built. So it collects the fragments from the PDB, and it uses about 25 fragments that are 17 residues in length and 25 that are 9 residue fragments in length. But I think you can define this yourself. And then they fit the fragment to the density. And they also look for the overlap with the rest of the chain. And so they fit that first to half map 1, but then they evaluate the fit to half map 2. And the idea here is that you prevent overfitting. And you select then the fragments that fit the density best, and also that have good geometry, and then you refine these against the full map. And it also performs magnification refinement, as I mentioned, so it can, if your microscope is poorly calibrated, it can adjust the map. And it also performs a B-factor refinement, which is the same also for Rosetta, for Phoenix, and for RefMac. Now, B-factors should correlate with the local resolution. So you can compare your model colored by B-factors with local resolution just as a kind of sanity check to make sure that what you're doing, what the refinement is doing, matches what your local resolution looks like. And this really is a form of validation, which is comparing what you've done with what you expect to have happened. And really, with model validation, we need to consider 3 questions, which is, does my model really fit the map? Does it only fit the map because I've simply placed atoms everywhere that I see, density, and does my model look like other structures? And is it consistent with what we know about chemistry, what we know about physics? And so, does my model agree with the map? And there's various measures of how well the model agrees with the map. RefMac uses FFC average, which is to calculate the FFC between the Fourier shell correlation between a model and a map, and it calculates it over resolution shells, and then it works out the average from these shells. And the important thing about this measure is that it is independent of the blurring B-factor. So if you look at the FFC before refinement, you can see that it doesn't change no matter how much you blur your map. In contrast, the R factor, which is kind of the normal way in which you measure fit to map in X-ray crystallography, is really an inappropriate measure because it is affected by the blurring factor applied to your map. So you can see that before refinement, as you blur your map, the R factor falls, which is not really what you want. But also, as well as these global measures, we need to consider local measures of how well your model fits the map. Often, we're not interested in the whole molecule, perhaps part of the molecule, or a particular domain or a particular region. And so we can calculate FFC average, real space correlation coefficients, for just a particular region of the model, or we can calculate them for each residue in a chain. And this can help to identify problematic regions, regions that do not fit the density particularly well. And each of the 3 packages have methods for plotting these kind of graphs. And next, we need to consider, have we overfitted our map? Have we started to build into noise? So here you can see a model that nicely fits the data and a model which overfits the data. When you have an overfitted model, you can no longer predict what the next point will look like. And so to do model validation, we could reserve a whole half map for validation. But that is quite costly. We really want to include that information. And so we developed this system where you take your model, you refine it against your full final post-processed map, and from that you generate your refined model and also the refinement parameters that you used to generate that refined model. You then take your refined model and you scramble the atoms inside that model. So you add displacement values to each of these. And the idea is to shake it up to remove any kind of model bias. And then you refine that shaken PDB against just one of the half maps using the refinement parameters that you used for the full model. And then you compare that refined model both against the map it was refined against and the other half map that it hasn't seen to calculate 2 different FFC curves. And then you overlay these FFC curves. Now, if there's no overfitting, if you've not modeled noise, and noise will be different in both of the half maps, these FFC curves should overlap. And also, they should maintain predictive power. So beyond the resolution cutoff that you use during refinement, there should be a nice gentle slope down to near zero. In contrast, in an overfitted model will fit the map that is refined against far better than the map that it's never seen before, because you've modeled into noise which is specific to that particular map. And so you'll see this divergence between the FFC curves, and also you'll notice a sharp drop at the resolution cutoff limit as it loses this predictive power. As well as considering how well our model, because we can build a good model that fits the map, but does that model actually look like how a protein structure should look like? And this is one of the few validation packages that really tries to assess that particular question. And it determines the compatibility between your one-dimensional structure, your sequence of your protein, and the tertiary structure, the three-dimensional structure that you've built. Does a leucine particularly like to be on the surface of your protein? And this is very good at identifying regions that you've mistraced and misbuilt. But one of the standard ways in which we assess model quality is through the Molprobity package from David and Jane Richardson at Duke University. And this provides kind of a multi-criteria check on the geometry inside your protein or nucleic acid. And it's available both as online and through the Phoenix package. And you shouldn't just wait until the end of model building refinement to run this package, but you can do it as you're kind of building. And one of the nice things that it does is it looks at potential hydrogen bonding networks in your model, so it can identify nucleotides that perhaps need to be flipped to really maximize hydrogen bonding. And it also generates this table of statistics. So it looks at things like, are atoms overlapping in the way that they shouldn't? Do you have good rotamers? Is your Ramachandran plot okay? Are your bond angles and bond lengths close to what we expect? So this is for the Molprobity report for the highest reported resolution structure by Cryo-EM. So you can see that again operates on this traffic light-based system, but here perhaps the clash score is worse than you might expect for a structure at this particular resolution. And then you can look at those clashes in COOT. So again, showing how validation and model building are interconnected. So you can look at the clashes in COOT and perhaps identify on the left there where a side chain isn't perhaps in the right place, and then on the right here where a proline is clashing, and that's perhaps because the main chain needs to be optimized and fit in density. So validation can really help inform earlier steps of the process. The Ramachandran plot has been used for decades in X-ray crystallography and remains one of the best ways of assessing protein quality. And the idea here is that dihedral angles in your proteins can only adopt certain conformations. And so you can compare the dihedral angles in your protein with those that are energetically favorable to identify outliers. And different secondary structure occupies different parts of this plot. Now, as the resolution decreases, you do tend to see more Ramachandran outliers because it's much harder to fit your model into the map. But really, the aim here is to obtain zero outliers, as few as possible, although some that are outliers may be real because of the environment around them. Now, Molprobity and also the PDB, they don't just use a single Ramachandran plot, but they use multiple ones because different combinations of amino acid can result in different Ramachandran plots. For example, those near a proline are much more restricted in the conformational space that they can sample. And so this is why your Ramachandran statistics can differ between different programs. And then EmRinger. So EmRinger is really one of the very few validation metrics which is specific for cryo-EM. Most of the others are also used for X-ray crystallography. And this was developed in James Fraser's lab. And the idea is that you take advantage of the highest resolution information that you have in your map, which is around the C beta position, so where the side chain starts. And so it takes your C gamma atom and it rotates it around the chi-1 angle. And each point in this rotation, it interpolates the density of the map and then plots that in this blue line here on a graph. And we know from other high-resolution structures that the chi-1 angle is rotameric, and it usually exists around 60, 180, or 300 degrees. And so what does it mean when your peak in the density doesn't match one of these rotamers? It perhaps means that your side chain isn't modeled correctly, but actually it can more often mean that your backbone isn't modeled correctly, and that you've had to place the side chain in a region it doesn't really want to be because the backbone isn't correct. And so you can calculate an EmRinger score, which correlates with resolution. And although there's no real target EmRinger score to obtain to, this graph gives you a good indication of what kind of score you should be aiming for at which particular resolution your map is.
Does the movement of 5778 indicate
so
the replacement of the backbone?
So the movement of 5778 was in their paper, which they then took and then refined, and then showed that as you refined that particular model, then the EmRinger score would improve. And then the PDB also, before you deposit your model, will also perform some validation calculations. And it will give you tables of statistics. It will analyze every single residue in your model, and it will compare your model to all other structures and to all other EM structures. So you can see whether what you're doing is as good as what other people are capable of doing. And validation statistics should always be presented in your manuscript as a table that looks like this, more or less. There's no standard yet for how this table one should be organized and which metrics to include, but there should always be some kind of measure of how well your model fits the map, and whether that's correlation coefficient or FFC average, and mole probity statistic for Ramachandran outliers, similar statistics for nucleic acid geometry. And once you're happy with everything, your model looks good compared to other structures at similar resolution, then it needs to be deposited. It's now deposited together with the EM maps. And while you're depositing your model, I suggest that you also deposit the final map that you use to refine your model against, both half maps, which you should use for the validation, any masks supplied during post-processing, and any map that has been modified in any way, but not necessarily including sharpening and blurring, as long as you describe what you did. But any map that you may have modified should also be deposited. So in conclusion, I hope that I've shown that when you first get your map, you may think it's just one big unknown. But really, it's a Rumsfeldian ensemble of known knowns, known unknowns, and most tantalizing of all, unknown unknowns. And really, the purpose of model building is to maximize your knowns, to minimize your unknowns, and then to convert the unknowns into knowns. And then the purpose of refinement is to make your knowns and your now known unknowns look more like other known knowns. And finally, the purpose of validation is to compare your known knowns with your now known unknowns to other known knowns to find if anything that you think you know is actually still unknown. Honestly, I don't know why I do it. And before I confuse myself further, I'd just like to acknowledge the software developers, many who I have just oversimplified their programs massively, the institutes that hire software developers, the funding agencies that fund software developers, and everyone I stole slides from, especially Rob Nichols. Thank you for listening.